Okay, can somebody confirm you can hear me?
Yes, I can hear you.
Thank you. Let me share my screen. Let me shrink that down a little bit. All right, before we begin, are there any questions? Oops. Okay, today is January 15th, and we're going to try and finish chapter 1 today. We should be able to do that. Today will be a short lesson, and after chapter 1, I've decided we will not go further today. In the lab today, I think I will just discuss and begin the infectious disease project, and then that's it for the lab today. Any questions? All right. So last time, the slide we talked about was a history of microbiology, and we mentioned that in the fossil record, you can go back 3.5 billion years and find the first fossils of the ancestors to bacteria. And that was about 3.5 billion years. This will be the only date you need to know in the history of microbiology. I can't recall any other dates you need to know in microbiology. So this might be the only one, but certainly in the history of microbiology, this is the only one you'll need to know for the quizzes and exams. All the other dates, I'll mention it just to sort of give a I don't know, chronological order, but you don't need to know the dates for all the other parts for the history of microbiology. You do kind of need to know the relative order, like before the Mayans could record making a fermented beverage from cacao, which we'd call close to chocolate. The Babylonians had to write down their recipes for making beer, and you had to have the first fossil evidence of life on Earth, so that this knowledge of the Mayans had to come after the preceding knowledge. But you do not need to know the dates. In 1590, you don't need to know that date. It's another important event in the history of microbiology, and that's when Janssen developed the compound microscope. Microscopes were not used in biology for a long time after he invented it. And to tell you the truth, I don't know what the early people used microscopes for, but it was not for looking at even biology, even cells. The first use of the microscope for looking at cells or biology happened by Robert Hooke, who reported using the microscope. And when he looked at living things, particularly plants, he found that they were composed of little boxes, which he called cells. And let me see if I can draw that for you. What he did was he took shavings of cork and then looked at it under the microscope, and he noticed that there were cells, or I should say boxes like this, in the cork. And by the way, when I'm saying cork, I'm referring both to the cork tree and to the bark of the cork tree. And then cork is used also to, I don't know, plug wine bottles. And they usually use cork of the cork tree to make the cork that they plug the bottle with. Any questions about the term cork? And then this would be repeated. I'm not going to draw all of them. And this reminded Hook. Let me get an eraser here. Erase this part right here. This reminded Robert Hooke of the rooms in a monastery where the monks would have a private room. Let me get this back. And they'd have like a door into their room. And this would be the hallway. And then this would be the rooms of the monks. And they called these rooms cells. And this reminded Robert Hooke of that. And so that's where he termed the word cell from. So the cells in living organisms come from the rooms in a monastery, because the first cells Robert Hooke looked at, the plant cells of the cork tree, reminded him of the cells of a monk. Any question about any of that? Now, Robert Hooke did take a look at animal cells, and other microbiologists did, but the compound microscope was not developed good enough that it could find focus good enough. So everything looked early in this day blurry. And if you looked at the tissue of animal cells, you could not see the cells. But when you looked at the tissue of a plant organ, plant tissue, you could see the cells in plants. And why do you think they could see the cells in plants, but not the cells in animals with the early microscopes that were blurry? Okay, what do plant cells have that animal cells lack? Come on, I know somebody knows that.
Cell walls.
Yeah, the cell wall is very thick and rigid, and all plant cells have them. Animal cells lack them, and the cell membrane could not be seen by the early microbiologists or the early biologists. So people were not able to see cells in animal tissue with the early microscopes, but they could see cells in the early microscopes with plants because of the cell wall. Robert Hooke's discovery of cells in plants marked the beginning of the cell theory. The cell theory is that all living things are composed of cells. And this theory was developed by others. For example, Virchow was the first person who actually wrote it up and then published the cell theory. All of the above events that I've talked about are not specifically microbiology. These are things that happen in the history of microbiology before people looked at microorganisms, meaning the cells that Robert Hooke looked at were not microbes, they were plants. The history of microbiology was largely concerned with the hypothesis of how life originates. There were two competing hypotheses to explain how life began. And this argument happened between the 1600s to the 1800s. And that is, all life came from spontaneous generation, where life arises from non-living organic matter, or I should say not organic matter. And what happens is a vital force comes in, and then life spontaneously generates. The other theory or hypothesis was that life occurs through biogenesis, and that is life arises only from pre-existing life. Even in the early days, people would agree that if you were talking about where does a baby come from, that it came from its parents. And so when they were talking about complex organisms such as a baby, everybody agreed that the baby came from pre-existing life. However, they would argue that other life forms could spontaneously generate. There were beliefs supporting the spontaneous generation, and there were things like if you threw a log into the fire, a newt would crawl out of the fire. And people would argue, wood in the fire, the wood together with the fire and the vital force comes and generates a newt, and then the newt crawls out. And the flies emerging from manure, the same thing. And then maggots arising from a decaying corpse, the same thing. Most were logical, but none were scientific, and some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar and put wheat kernels in the bottom of the jar, it would spontaneously give rise to mice. In reality, the mice climbed into the jar to get the wheat kernels, and then because the jar had the steep edges, the mice couldn't climb out, and the sweaty underwear had nothing to do with the arrival of the mice in the jar. Was somebody going to ask a question? All right, somebody got your microphone on. Please mute it. Now, one of the reasons why microorganisms were said to spontaneously generate was you could see microorganisms starting to grow almost everywhere you looked early on in the history of microbiology. Normally, when you would make a tube of media, it would normally look like this. It'd be clear with no growth in it. And then if you left the tube out, it would become turbid like this tube. And that was from the microorganisms growing there. Somebody needs to mute your microphone. Let me see if I can figure out who this is. They must have muted. What was actually happening was there'd be microorganisms in the tube or in the media, and then when you make the media, they would then grow in the tube. And even if you sterilize the media and then put it into the tube, microorganisms could fall out of the air and then grow in the tube, making it turbid. The first evidence against the theory of spontaneous generation came from Francesco Redi. You don't need to know the date, but it was in 1668. And this is actually an important experiment that was the first controlled scientific experiment, at least the first one that we know about because it was written up and published. What Francesco Redi did was he got 3 jars and he left them open and he put meat in them, and maggots appeared in the meat. And then he got 3 jars and he completely sealed the jar and they got no maggots. And the people who believed in spontaneous generation told Francesco, no, that won't work because you sealed the jar, the vital force couldn't get in, and that's why there were no maggots generating. And so Francesco Redi did something ingenious. He covered the jars with a fine wire net, just a metal net. And he then left the meat in there, and the vital force could get in through the netting, and no maggots arose. So from his experiments, we had the first evidence that at least maggots could not spontaneously generate. And after Francesco Redi's day, everybody agreed that complex life forms, like most eukaryotes, could not arise by spontaneous generation, that they had to arise by the theory of biogenesis. But people would argue that the microbes could arise by spontaneous generation, because if you put the fine wire mesh over the jar and then you made media, the microbes would grow in the jar. Now, the first observations of microbes actually happened by this guy here, Antony van Leeuwenhoek. And he lived from 1673 to 1723. He was the best microscope builder of his day. And he also had very good eyesight. And he was the best microscope builder of his day because he was the best person at grinding lenses. And when he made his microscopes, he used them to look at a large number of things. And he described living microorganisms in scrapings from his teeth, in rainwater, in peppercorn infusions. And when he sampled the River Thames, the water supply for the city of London, he discovered tads of microorganisms. This is actually a picture of one of his surviving microscopes, and it was rather simple by our standards, even simple in the terms of a microscope, and that it was not a compound microscope. It was a simple microscope. A simple microscope only has one magnification lens, and so really all it is a super duper, what do you call that? Drawing blank on
A magnifying glass?
magnifying glass. But he actually ground his own lenses, and they were so good that he was able to clearly see through them. And as I mentioned before, when you had the compound microscope with two lenses, they were blurry and people could not see cells of animals. I don't know if Antony van Leeuwenhoek ever looked at animal tissue, but he did look at microorganisms and found them on almost everywhere. And he saw some cells that were so small they were not seen for almost a century. And here is his picture here. And if you don't know, that is a picture of a bacteria. That is a bacilli, and that's the movement of the bacilli in his drawing. And there's a spirochete he drew. Figure E are cocci. And then here's again some more bacilli. That one is either two bacilli or a bent bacilli. Like I said, he was so good at building microscopes, he was able to see bacteria, cells so small that other people could not see them for nearly a century later, What happened was, after a century, people got better at making and grinding the lenses to a compound microscope. And so a century later, we finally had a compound microscope that was good enough to see bacteria. All right. So you should know that Rudolf Virchow is the person who wrote up the theory of biogenesis, stating that all living cells can arise only from pre-existing living cells. And this became known as the concept of biogenesis. However, even after his day, 1858, when he published this, there were still arguments about whether microbes could spontaneously generate. And that argument continued until 1861, when Louis Pasteur gave the first evidence that disproved the spontaneous generation theory and confirmed the theory of biogenesis. First, you need to know that Louis Pasteur also discovered that microorganisms are present in the air. And that's why when you made a tube of media, even if you sterilize the media in the tube, if you left the tube open, microbes would fall in from the air and then they would grow in the tube. But Louis Pasteur showed that microbes can be destroyed by heat. And then he did his ingenious and famous swan neck flask experiments to disprove the theory of spontaneous generation. What he did was he made media, put it into a flask with a long neck, and then he heated the flask right here, making it a swan neck, where it was open to the outside for the vital force to get in. But when he left it, he saw no microbes growing in the media here. You have to heat the media to sterilize it and then let it cool, and then no microbes would grow in. What happened was the microbes from the air came down to the tubing right here, but the microbes did not go up against the force of gravity. And so to get the growth in the media, you've got to make the media come back to here and then move the media back into the flask. And this disproved the theory of spontaneous generation. All right, beginning with Pasteur's work, there were a number of discoveries, including the relationship between microbes and disease, and this relationship became known as the germ theory of disease. The different germs or bacteria cause disease. And I guess I should say microbes, because there are many microbes which are not bacteria that can cause disease. Out of this work came the theory of immunity, that the immune system could protect you from microbes causing disease. And also out of this work came the work of the antimicrobial drugs. Also out of this work came Pasteur's concept of aseptic technique, where you can get a culture containing only one species, and then if you use aseptic technique, you can keep it sterile, or not sterile, but pure, so that there's only one species growing in the media. And how you do that is you just limit the exposure to the air and sterilize the loop or the tool you're using to transfer the media from one container to another. And that's aseptic technique, which you'll learn about in the lab. Koch, at this time, developed the technique for streaking for isolation on a solid media. And Louis Pasteur did come up with the idea of getting a pure media, but it was very difficult to get the pure media, what he did was he did limiting dilution, and he would make replicas of the plate and different dilutions, and then he would plate each of the dilutions into 10 tubes. And if only one of the 10 tubes had growth, 9 of the tubes were empty, he would say the one having growth was likely colonized from one cell, and therefore it would be a pure culture. And that was true, but it was a very difficult way to get a pure culture. Robert Koch came up with a much easier way, and that is you just streak for colony isolation on a solid media. And if you streak 2 times, meaning you get a single colony and then streak it out again a second time and get a single colony, the cells from that single colony that went through 2 colony formation events is highly likely to be a pure culture. Any question about any of that? All right, about the same time, Hess, this is a co-worker of Dr. Koch, somebody who was working in Dr. Koch's lab, developed nutrient agar, a new and very good solid media for growing microbial growth. And about this time, Sergei Winogradsky developed his idea that microorganisms were involved in the biogeochemical cycles, like the nitrogen cycle, how the microbes move nitrogen in one part of the ecosystem to another, changing the form of nitrogen. About the same time, Louis Pasteur also came up with the concept of pasteurization and fermentation. Louis Pasteur noticed that alcoholic fermentation was the conversion of sugar into alcohol that was used by the beer and wine industry to make beer and wine. And he correctly concluded that microbes were responsible for the alcoholic fermentation, that microbes were fermenting the sugar and making the alcohol. He also correctly concluded that when the beer or the wine became spoiled, it was because there were other microbes growing in the beer or in the wine, changing the alcohol and fermenting it into vinegar or acetic acid. And then the beer and the wine would not be very palatable and no one would want to drink it. The beer and the wine industry actually asked Louis Pasteur to help them figure out how to stop the beer and the wine, I think it was the beer industry, from spoiling and changing from beer into a vinegar-like drink. And Louis Pasteur came up with pasteurization, where the spoilage bacteria could be killed by heating, but the heat was not hot enough or long enough to evaporate all of the alcohol in the beer and the wine. And this process became known as pasteurization after Louis Pasteur's name, application of high heat for a short period of time. Pasteurization is used in a wide variety of foods. Most people associate it with pasteurized milk, but it happens in a large number of other foods, including beer today. Most beer is pasteurized. The exception is Coors beer, and they filter Coors beer instead of pasteurizing it to remove the microbes. All right, the concept of fermentation, that microbes could affect an organic material and change it from one chemical or molecule sugar into alcohol, led people to ask, Could microbes have a similar relationship with plants and animals? And might microorganisms cause disease? This idea became known as the germ theory of disease. There was no one person who put forward or developed the concept of the germ theory of disease. It was a concept that grew out of many different people's ideas. Originally, people believed that diseases came from the breathing in of poisonous vapors. So when you went walking through a swamp, people believed you breathed in the poisonous vapors. And then if later you developed cholera, it was from breathing in the poisonous vapors of a swamp. In reality, the cholera bacteria was in the swamp, and then the people simply breathed it in, or they touched something and then ate something without washing their hands, and then they got the cholera. All right, let's skip that. You can read about that. Robert Koch was the first person who proved the theory, the germ theory of disease. Now, note, I'm not saying he's the first person who proposed this theory. He proved the theory. And what he did was he took the bacterium Bacillus anthracis and then showed that it causes the disease anthrax. And then he provided the experimental steps that he used to prove the Bacillus anthracis causes the disease anthrax. And these are the steps that we use to prove that any microbe causes a specific disease. And these steps are called Koch's postulates. The steps you use to show that a given microbe causes a given disease. This proved, or Dr. Koch proved, the germ theory of disease. And then he published the steps he used, and they became known as the Koch's postulates. Around this time, Edward Jenner developed the first vaccine, starting the first vaccination. The first vaccinations were against the disease smallpox. What Edward Jenner did was he inoculated a person with cowpox. And then the person who was inoculated with cowpox was protected from getting smallpox. No one understood how Edward Jenner's vaccine worked. And it was not understood until some work that Louis Pasteur did, where Louis Pasteur showed that what happened was cowpox was a virus that was similar to smallpox. And then when the immune system reacted to the cowpox, some of the immune system cross-reacted to smallpox. And so if you became immune to cowpox, you were then immune to smallpox. And after Louis Pasteur's work, people were able to figure out how to make other vaccines. The term vaccination is actually derived from the word vacca, which is the word for cow, after the first vaccine, which used the cowpox virus. Any question about that? Let me just briefly mention that cowpox was a disease of cows, and milk maidens who milk the cows frequently came down with cowpox. And there was a what do you call it, a wives' tale or a woman's tale about milk maidens, that if they got cowpox, they were immune to getting smallpox. And one milk maiden suggested that to Edward Jenner, and he looked into it and then actually gave two boys, one of them was his actual son, cowpox. And cowpox was a very mild disease in people. They usually only got one, I don't know, blister or skin lesion, one small, like mosquito bite from the cowpox. And then he later checked to see if they came down with smallpox. And I think what he did was he just waited until smallpox hit the town or the city. And then he noticed that the two boys who were inoculated with cowpox never came down with smallpox. Edward Jenner had no evidence that cowpox would work. He just believed it. And then he had no evidence that cowpox wouldn't hurt the boys that he was inoculating. He just believed it. And that would be unethical today. Experimenting on children, especially on children, but just pointing out to you that Edward Jenner would do things that people would object to today. The protection that people got from the cowpox virus was known or called immunity, meaning the immune system protects you from the disease smallpox, which, if you don't know, is a very serious disease. Smallpox is currently eradicated from the world. All right, let's develop this work a little further. And out of all of this work came the concept of chemotherapy, that you could treat a disease with chemicals. And how this came about was once you showed that infectious diseases were caused by a microbe, people got the idea maybe we could find a chemical that could cure a disease. And chemotherapeutic agents were used against infectious diseases. And people came up with the concept of an antibiotic or a synthetic drug to treat diseases. Now, let me mention that chemotherapy today is largely used for treating cancer. But originally, it was used for the treatment of any disease, including microbial disease. And it can be used that way today, but most of the time chemotherapy is used exclusively to mean the treatment of the cancer disease. Any questions about any of that? So I won't use the term chemotherapeutic drug for treating an infectious disease. I'll use the term antimicrobial drug. You can have different antibacterial drugs for treating a bacteria causing disease. And there are two types of antibacterial drugs. There's an antibiotic, which is an antibacterial drug that is made by a living organism, mostly a microbe, such as bacteria or fungi. Penicillin G would be an antibiotic. And then the other type of antibacterial drug is a synthetic drug. This is a man-made antibacterial drug that works on bacteria. And for textbook authors, it's important to make the distinction. The trouble with the distinction is penicillin G is an antibiotic, and carboxycillin, is another member of the penicillin family, is not an antibiotic. It is a synthetic drug, a man-made drug. Now, that's the textbook definition. However, the way antibiotics are used, the term it's used in the common usage in like the American public, and even the common usage among doctors, the term antibiotic applies to any antibacterial drug. And that is generally the term that I'll use when I'm using the word antibiotic. I usually use it to mean any antibacterial drug that can work against a bacteria. But you should know your textbook does not use it that way. The first antimicrobial drug that was ever used was the bark of the cinchona or I used to know how to say that, cinchona tree. Both cinchona and cinchona are correct pronunciations. And the bark of this tree happened to have quinine in it, a chemical that is used even today to treat malaria. And then Paul Ehrlich found the first chemical that was successful at treating the disease syphilis. And he found this chemical in 1910, and he called it a magic bullet, a chemical that could prevent disease by an antibacterial agent. And what he found was salvarsan, a synthetic arsenic drug that could kill syphilis, and it was toxic to people, but if you applied it at the right dose, it could cure syphilis and not kill the patient. Any questions about any of that? All right. When do you think quinine was first used in Europe to treat malaria? Anyone have a guess? Well, was it World War II? Around that time, 1939 to 1945? Was it around 1910? Was it sometime earlier in the 1800s? Sometime before that? Or maybe sometime later? What's your guess? Come on, people, take a guess.
1800s.
1800s is a good guess, but you're actually wrong. It was actually in the 1600s. I forget the exact year because it's not known, but I think it was the mid 1600s. What happened was Jesuit missionaries working in Peru noticed that the native Indians were eating the bark of the cinchona tree to treat their malaria. And the Jesuit missionaries noticed that it was successful. And then they sent the bark of the cinchona tree back to Europe. And then the bark was the first treatment that Europe ever had for treating any drug. And like I said, that was the middle of the 1600s. As a side note, you don't need to know this, but I'll mention that some doctors actually gave patients malaria. And the reason was the high fever of malaria could cure certain diseases. And this was one treatment that was actually successfully used, I might add, to treat certain diseases like syphilis. And somebody who had syphilis, you could give them malaria. The high fevers of malaria would cure the patient of syphilis. and then the doctor would fall back on the cinchona bark to treat the malaria. This treatment was risky, and that is, you might fail to cure the patient of malaria, or the malaria and the other disease could kill the patient. But it was a successful way of treating disease before the advent of antimicrobial drugs. As I stated, Ehrlich came up with the first chemical to cure disease in 1910. You don't need to know the year there, but you should know that he came up with a drug to cure syphilis. And then the first antibiotic was discovered in 1928. Once again, you don't need to know the year, by Alexander Fleming. And what he discovered was he streaked out a plate of bacteria, and he was a sloppy microbiologist. And then he just left the plate on the counter, and mold had gotten into the plate. And then he came back after the weekend and found the plate on the counter and saw that there was bacteria growing on the plate, and then a mold colony like this. And when you look at it, you'll note there's no bacteria in this region of the plate here. And Alexander Fleming correctly concluded that coming out of the mold colony was a chemical that was preventing the bacteria from growing. And this was later called an antibiotic. And the first antibiotic, Alexander Fleming called it penicillin, after the mold, which is penicillium with an M. And then the antibiotic is called penicillin with an N. Alexander Fleming experimented with penicillin, which is actually penicillin G, but he was never able to purify it any better than simply getting media that he was growing the mold in. And so there'd be mold spores in it and other stuff in it, and he could never purify it further than that. So penicillin G was never used until the early 1940s. It was in World War II when penicillin G was first purified and then used in the clinic. So it was not the first antibiotic to first become clinically available. The first antibiotic that became clinically available was a sulfa drug, sulfonamide, and why it was easier to purify was it was man-made, and it became clinically available in 1935. Once again, you don't need to know the year, but it beat penicillin by a little more than 5 years to become clinically available. I'm talking about penicillin G. All right, let's talk about microbes and human disease. Microbes were once classified as plants, hence the term flora for microbes. And if you ever hear me use the term microflora, I'm sorry, that's the old term that I was taught when I learned microbiology, and that was a long time ago. And that was long before the term microbiota was ever invented. And microflora is an old term. Flora refers to plants. And we use the term microbiota now. And normal microbiota are the microbes that are normally present in and on a person. And here we would call that the human normal microbiota. It's been observed that all animals and plants host a symbiotic microbial community where there are many different microbes growing in and on the animal and plant, and each species has its own normal microbiota. For example, humans have E. coli in their gut, and we do not have salmonella in our gut, but birds have salmonella in their gut. It's part of their normal microbiota. And then humans don't normally have Bartonella henselae. It's part of our normal microbiota, but cats do. And Bartonella henselae is a bacteria that causes cat scratch fever. The normal microbiota is actually central to the welfare of the plant and the animal. The normal microbiota are usually found on and in the skin, the oral and nasal cavities, the respiratory tract, the digestive tract, and the urethra. The normal microbiota is not normally found internally in internal organs and tissues. The normal microbiota can prevent the growth of pathogens. What happens is the normal microbiota competes for space and resources, preventing the pathogen from getting established and then causing disease. The normal microbiota can also secrete toxins that inhibit the growth of pathogens. And the normal microbiota can produce nutrients and growth factors for their host. For example, for humans, our normal microbiota help make folic acid and vitamin K for us. We get a significant amount of both folic acid and vitamin K from our normal microbiota. And then lastly, the normal microbiota is helpful in the digestion of our food. All animals have a normal microbiota that helps them digest their food. Any question about any of that? All right. The normal microbiota does not normally cause disease. However, it can cause disease in certain circumstances. Like if the normal microbiota is growing on an AIDS patient. An infectious disease is a disease that happens when a pathogen, a microbe that can cause disease, overcomes the host resistance and then grows in the host, causing the disease. And we call a microbe that can cause disease a pathogenic microbe. Remember that most microbes are not pathogenic. An emerging infectious disease is a new or changing disease that is either increasing or has the potential to increase in incidence. You should know what the definition of an emerging infectious disease is. And can anyone mention a fairly recent emerging infectious disease? All right, I want to end this, so I'm going to fill in the answer there. COVID-19 is an emerging infectious disease because only a few years ago, COVID-19 had never hit the human population. Another example is the bird flu. Could be an emerging infectious disease. Some people are worried that it could change, and then when it infects people, it could then spread from people to people. As it is now, the bird flu only spreads among people, but birds can give it to people. But once the person gets it, it doesn't spread people to people. And if it ever were to spread people to people, it would then become a new infectious disease. So that would be an emerging infectious disease. Any question about any of that? If not, I've got more slides on emerging infectious disease. However, I'm not going to go over it, nor am I going to quiz you on it. I'll only quiz you on what the definition of an emerging infectious disease is. Any question on any of the topics of chapter 1 and chapter 10? All right. If there's no questions, I'm going to end the lesson here. And
Can we start the lab earlier, maybe today?
no, because there might be people showing up only at 6:30. But I
Okay.
promise the lab will not be very long today.
All right.
Did I say I was going to have the lab today?
You said you just wanted to go over the infectious disease project?
Okay. All right. So we'll do that at 6:30. Sorry, but we do have to wait until 6:30.
Okay.
All right.
All right. Thank you.
All right. Bye.
Bye.